Welcome Genetic Teacher channel. Today, we are talking about bacterial binary fashion, in form of bacterial asexually reproduction, into typically two daughter cells, using conjugation method, by exchange bacterial plasmid DNA. Firstly, bacteria copies DNA to each other, then, bacteria transfer their genes by bridge, which made of plasmid connect to ends of two bacterial cells. After that, conjugation transfer genes that code against antibiotic, which help bacteria fight off antibiotic, as mentioned in next slides. And the question is, why conjugation method? And the answer of this question abbreviate into 3 points. The conjugation allow DNA to be transferred from one bacterial cell to another, and genetic codes used to protect against antibodies, which help bacteria survive in normal conditions. And finally, let's define the differences between conjugation and meiosis divisions. As we showed, both send genetic materials to another cell, but conjugation create unique bacterial cells by exchange genes, use plasmid tubes, while meiosis divisions create completely unique copies of genetic information, use sexual reproduction. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.